Hello everyone. Today we're going to learn about EcoR1 and PST1. So first of all, what are they? They're both different types of restriction enzymes. So that's very important. So both, let me try. Restriction enzymes. And what are restriction enzymes? Well, restriction enzymes, so they recognize short, specific DNA sequences, typically 4 to 8 base pairs in length, and once they find these sequences, the enzymes know when to cut and cleave off the DNA segment. So I like to think of them as molecular scissors, right? Because I feel like a nice analogy. Scissors, they cut things. These also cut things, right? So, yeah. But now we know what they are. Let's look at the similarities and the differences between them. So, recognition site. So where it cuts. So for EcoR1, if the segment looks like what am I saying? 5 prime, G, A T T C. It does continue, but in the 3 prime, if it sees this, boom, a cut right here. So for PST1, also goes 5 prime, 3 prime direction. Everything does. And just to make it clear, this is Roman numeral 1, not I. Well, looks like an I, right? That's why I call it 1 and not PSTI. But never mind. So yeah, so it was 5 primed, C T G C A, 3 prime direction. But it's interesting. It cuts. Oh, I forgot to add one. Sorry. G 3 prime. And it cuts right here. So this is how the recognition sites for both of these restriction enzymes look like. Next thing, type of cut. So for EcoR1, it's a 5 prime overhang. So it cuts at the beginning of the sequence right here. And that's the type of cut. And then for the PST1, it's a 3 prime overhang. And it cuts at the end of the sequence right here. So palindromic, you might have heard the word palindrome before in English class, not my favorite class, but still. A palindrome is when something is spelled the same forward as reverse. So for example, race car is spelled RACECAR, forward, and backwards RACERCAR. So that's a palindrome, and that's how those work. So these restricted enzymes cut at palindrome sequences. All restricted enzymes do that. So they are both palindromic. So this full sequence for EcoR1 goes GAATTCCTTAAG. So you can see it's the same forward as the back. And then this is the middle where it can almost be like mirror imaged or superimposed, right? And then for PST1, it's also palindromic. So it goes C T G C A G G A C G T C, and just like the other one is palindromic, same forward and backwards. And that's very important in any time you're doing like molecular biology and stuff like that, just because if you see something that's palindromic, then you know, I would say 99% of the time, because I don't know fully, but a restriction enzyme can be involved. Okay, next, optimal temperature. They both work optimally at 37 degrees Celsius. That's quite self-explanatory. And common use, so the most common use, I'll say PCR. If you're getting specific fragments, you can look at them and compare DNA sequences and stuff. But it's also used for general cloning, I guess, for both of them, cloning. And a lot of molecular biology, I would say, has implications of both of these restriction enzymes. That's how I learned about it as well, just like mostly in the lab and stuff. But yeah, these are the similarities and differences from EcoR1 and PST1. Thank you for watching.